Welcome to the third video of the first section of MMP. This video will include carbohydrate metabolism, glycogen regulation, the pentose phosphate pathway, as well as gluconeogenesis. So in humans, there's 4 types of glycosidic bonds we can digest. We have alpha 1,4. This is what you'll see in starches a lot. You'll have beta 1,4. These are in things like lactose, so the sugars you get in milk. You have alpha 1,6. These are also found in starches. They kind of branch starches together, and we'll talk a little bit more about that later in this lecture. And we also have alpha 1,2, which is in sucrose. So your things like table sugar. Now the way we break down macromolecules of sugars, like starch, as you can see, there's a lot of bonds here. And these are alpha 1,4 bonds, as I mentioned before. The way we do that is with an enzyme called amylase. Amylase is found in 2 places primarily. It's found in your mouth, so you actually begin digesting starches the second you consume something, as well as in your small intestine. So that will continuously break up the alpha 1,4 bonds and break it into smaller sugars. There are other enzymes. These are pretty much all found in the small intestines for the other types of sugars. So we have one for sucrose, one for maltose, one for lactose. And as for lactose, the lactase enzyme isn't always expressed in people as they get older. And the way that something like a lactose intolerance develops, for example, is without this enzyme being as present or as expressed, it's actually the bacteria in your gut will break down the sugar instead and produce gaseous products. It'll also draw a bunch of water into your intestine. And this will lead to a bunch of indigestion and gut problems. So that's how something like a lactose intolerance comes to be. And now, how does glucose actually get across membranes? So the exchange of glucose across membranes is actually passive. It's too big of a molecule to be directly absorbed across the membrane. However, we have these transporters right here. These are just simple channels, so these will work by diffusion. Something else we have, I'd say this is a little more common, is the glucose-sodium cotransporter. This is another passive process. The way this works is, let's say the diffusion gradient isn't, what's the word I'm looking for? It isn't favorable for glucose to move. Let's say there's way more glucose in the cell than on the outside of the cell. The way this can work is we can actually abuse the gradient. Let's say the sodium gradient is in our favor. And the way we can get that in our favor is by pumping out glucose to the cell with this potassium sodium ATPase. And what this essentially does is this uses ATP to get a gradient of potassium and sodium. You might have seen this before in cellular depolarization to set that up. It's also used in that, but it can also be used to make this favorable pathway for sodium to enter the cell, and then glucose kind of tags along with it. So kind of like a dual purpose there. Next of these transporters, you don't need to know these super in depth. What I would say to remember though is these are all the kinds of glucose transporters. GLUT1 is going to be your primarily CNS transporter. This will get things across the blood-brain barrier. GLUT2 is in the liver. This one's a little more important. You'll probably talk a bit more about this one in class, as well as I'll mention a little bit more throughout these lectures. GLUT3 is for the cerebral spinal fluid. So another one that's in the kind of central nervous system, just this one specifically for the, let me rewrite that, the cerebral spinal fluid. GLUT4 is for your muscle and adipose. This is another really important one. You've also probably seen on previous lectures, we briefly mentioned this one. So this is really important for your kind of peripheral cells that need energy / store energy. And then GLUT5 is actually weird. It's actually not a glucose transporter, but a fructose transporter. I think you're usually asked that one on a test just to like see if you remember. So that's a good thing to remember, that GLUT5 is kind of the odd one out. So that's basically all the transporters and basic metabolisms of different sugars. Moving forward, we can now talk about glycogen. Glycogen is the primary way we store sugar and carbohydrates in our body for energy. And the way we store glycogen is it starts with this protein called glycogen. So essentially all these branches on the outside here, these are the actual glycogen molecules, but in order to like kind of get a starting point, we need this glycogen protein. So essentially, glycogen can be degraded, and the way it's degraded in muscle for energy is we convert it into glucose 1-phosphate first, which is just kind of the breakdown process, but then it's important. If you remember, glucose 6-phosphate does not have the ability to escape the cell, which is really important for muscles, since your muscles are going to be the cells that need energy essentially all the time. So that conversion is really important just so the glucose doesn't actually leave the cell. And then all the processes like glycolysis will occur. If you have anaerobic respiration, you might have some lactate form, but ideally you get a lot of aerobic and you get the CO2 and ATP primarily. The liver is similar. You'll originally go from glucose 1-phosphate to 6, but then glucose 6-phosphate can actually be converted just to glucose in the liver. So glucose that's stored in the liver as glycogen can actually escape and be supplied to other tissues that may need it. Usually muscles, but also just other parts of the body as well. So it's kind of like your primary glucose reserve. Um, and the way we actually build glycogen is we have this glucose 1-phosphate right here. That's kind of like our building block, but we actually need an energy carrier as well to add an extra phosphate in order to link them. And that's UTP. It's similar to ATP in the fact that it's a triphosphate molecule. But in this case, it's used instead to kind of mark these glucose to be linked together. So you can see there's like an extra phosphate than was originally there. And this is essentially the catalyzer for linking molecules together in glycogen. Okay, so now that we have that UDP glucose, we can actually use it to add on to glycogen or to make some glycogen. So you can see this enzyme right here, glycogen synthase. You can think of it like synthesis. It will add on glucose in an alpha 1,4 bond. So you can see there's nothing right here, but once this is added, we add something on. This can also do multiple at a time if they're already linked essentially. So you're basically just making a bond between two different molecules already existing, whether that's a chain or not. We also have 4,6 transferase. This can essentially create a 4,6 bond to make a new branch. Glycogen likes to be branched. I believe it branches about every 20 residues, I want to say. That's never going to be something you're asked, but just kind of a little fun fact there. So this can add more branches on, just so it doesn't get too long in one direction, because I think that may cause like weird folding, or it might just be too long to store. So ideally, you want to be about 20 residues. So now on the other end, breaking down glycogen, the opposite to synthase is phosphorylase right here, this enzyme. So these two are opposite, and this will essentially do the exact opposite of what we just did with synthase, in the sense that you will take off a chunk or one, and it'll become shorter. We also have this enzyme down here, 4,4 transferase. This can essentially move a branch to like a different branch. This is actually in both glycogen synthesis and glycogenolysis. Just if you want to make a branch longer in like a straight chain. You can think of it, it's also kind of similar to synthase in that way, but it requires a branch to already exist in order to do it. So it's kind of weird, but it is in both. And then the last one is, you can see when we use our phosphorylase, you're always going to be left with the one, it's called a glycogen core kind of deal. And the way we get that off is we need our own enzyme because it's a different type of bond, and that's alpha 1,6 glucosidase. And you can think of that as kind of the opposite of the transferase, the branching enzyme. This is essentially a debranching enzyme. And with that, that's pretty much everything to do with glycogen buildup and breakdown. I would definitely remember these enzymes. I think all of them are fair game except maybe 4,4 transferase. I don't think we ever got asked that, but you might need to know it. So this is definitely a slide I would touch up on. So now we're going to talk about how hormones regulate glycogen production and degradation. So we'll start out with insulin, which is right here. I know this diagram looks crazy. All you really need to know is that when insulin moves things up into the cells, it'll create a cascade in the cells that'll eventually become this PP1 is where it ultimately wants to be. And then when that gets activated, you can see activation is the blue here. I'll try and draw this in blue. So these blue lines, and inactivated in red, which are these. Once it's activated, we will eventually move to glycogen synthase, which, as you may remember from the last slide, is the thing that builds. Glucagon, epinephrine, and norepinephrine are kind of the opposites. They will inactivate this pathway by going to pKa and eventually down to glycogen phosphorylase, which will break down glycogen once it's in that pathway. And the reason there's some overlap here is because these are actually co-regulated. And essentially, when phosphorylation cascades in cells occur from something like CAMP or other mediators, some things will be active and some things will be inactive when they're phosphorylated. And I think the diagram on the right does a really good job showing that. So for example, when a phosphate is added to glycogen phosphorylase, that will put it in its active form, and this will break down glycogen. When this protein kinase phosphorylates glycogen synthase, it will actually become inactive. So one way you can think about it is if inorganic phosphates are high in the cell, that probably means glycogen is being downregulated, and then the opposite is true as well. But of course, that all happens because of signaling cascades, and it's pretty complicated. I just try and remember it in the sense that if sugars are going into the cells with insulin, it's probably building up, and if epinephrine and glucagon are trying to get sugars out of the cell, it's probably breaking down. So the two ways that muscle can activate glycogen phosphorylase, it's different than in some of your other like storage cells, is there's your traditional way, which might just be from your kind of norepinephrine or your need from ATP or epinephrine or glucagon. Kind of what we've talked about before. It'll all go through the system, activate phosphorylase kinase. But the other way that's kind of interesting is you may or may not remember from, I believe, Anatomy 2. Instead of having like an endoplasmic reticulum, muscle cells will kind of share them across like several, and it's called the sarcoplasmic reticulum. An influx in calcium in that, which can also occur from like a nerve impulse or like flexing, will increase calcium into the cell, which can also activate phosphorylase kinase, kind of leading to the same effect. I don't think you could ask that, but that's just another way it can activate in muscles, which makes sense, right? If you have a nerve impulse, meaning you're using your muscles, it will also activate this system because you need more glucose for energy. So that's kind of just like a fun fact. This was on the slides, but I don't think we ever got asked this, to be honest. All right, so really quick, let's talk a little bit about fructose and galactose metabolism. I'd say the main takeaway is that in order to turn fructose and galactose into like usable products for something like the TCA cycle, you do need to input ATP, which is also true for glucose, if you remember, in the preparative phase. You need 2 ATP net for fructose and 1 for galactose. The enzyme I'd really recommend remembering is aldolase B here. This is the rate-limiting enzyme in the fructose metabolism. And eventually your last product is going to be glyceraldehyde 3-phosphate, which will just go directly into glycolysis. You can also make fructose from glucose. It requires NADPH, which is an energy signaling molecule, or energy carrying molecule, sorry. And it also produces an NADH. So you input a little bit of energy and you get out a little bit of energy to get fructose, which obviously has different uses than glucose in the body. It just kind of depends on what the body needs more of at the time. Okay, now we can begin talking about the Pentose 5 pathway. This is essentially a pathway to generate both some 5-carbon sugars for things like nucleotides, as well as generating that NADPH, which if you may remember from the last slide we just talked about, that can be used to turn glucose into fructose. It's also used for some other things throughout the body. But essentially how this starts is we start in the oxidative phase. We go through a series of reactions here. We generate an NADPH when we begin with glucose. We go through a lot of these steps. They're not incredibly important here again, generating NADPH. We eventually end with this ribulose 5-phosphate down here. What this does is this has one of two paths it can essentially take. It can become ribose, which is directly used in RNA, and then deoxyribose in DNA, or xylulose. This one's more of an energy molecule. They can actually be interconverted in the non-oxidative phase, which we'll talk about here in a second. But I just say, remember that you start with glucose, so nothing new there. Your NADPH is what you're trying to generate throughout the whole thing. And your ribulose is kind of like where you branch off and can do different things. So essentially the net equation for our non-oxidative pathway is we can exchange between 2 fructose and 1 glyceraldehyde and 3 ribose or 3 ribulose, depending on the stage you're in. These can go back and forth, and this is your non-oxidative reactions that this equation's essentially showing. Up here is oxidative until we get to like right here, or I guess technically right here. But we can go back and forth between that. Like I said before, your ribose can be used in synthesis of like RNA. And then up here we have our NADPH we generated on the previous slide. NADPH, like I mentioned before, can be used to turn glucose into fructose, but something else it can be used for is also the glutathione system that mentioned, I think, in the first series of lectures with the electron transport chain. It can be used to essentially recharge glutathione after it has detoxified something or unradicalized something. So that's another use for NADPH. Okay, so now let's talk about alcohol metabolism. So the alcohol you consume will be ethanol in the body. Only two enzymes, they're both dehydrogenase enzymes. We have alcohol or ethanol dehydrogenase, as well as acetaldehyde dehydrogenase. 3 steps, ethanol to acetaldehyde and then to acetate. Acetate's nice because you can turn it to acetyl-CoA, which can go into the TCA cycle to make energy. You can also see here we are producing some NADHs, so that's really good energy-wise, but consuming too much alcohol can lead to, obviously, the feeling of nausea. Nausea comes from actually this acetaldehyde intermediate. There's a OTC drug called disulfiram, or maybe this one isn't OTC, but how it works essentially is it inhibits the acetaldehyde dehydrogenase transition into acetate. And acetaldehyde is actually the chemical that makes you feel nauseous when you're intoxicated. So the drug essentially works by making you feel really sick whenever you drink alcohol. So that's kind of a fun one. But obviously beyond just the nausea, too much alcohol can lead to other issues in your body. While NADH may seem like a really positive thing, having those energy carrier molecules, it does run the risk of blood acidosis is the main one, and there's kind of 2 pathways through this occurs. You have your conversion of pyruvate into lactate, and if you have really high NADH, essentially you will push forward this reaction to lactate, and lactate will convert into lactic acid. And while this does give you a little bit of energy, this was similar to the non-oxidative glycolysis. It will increase your blood acidity. Another way through this occurs is actually back in our TCA cycle, this conversion of malate to oxaloacetate. You may remember this has a really high energy threshold, and that's because it can like lead into beta oxidation through the malate pathway, as well as producing ketone bodies. And essentially, this produces NADH when it works. So if we have high NADH, this isn't a very favorable reaction, meaning more malate will go into beta oxidation, make more ketone bodies, which will lead to ketoacidosis, which will increase the acidity in blood. It can also, through high lactate, lead to conditions such as gout. So it's really not good for your kidneys to deal with this, as well as your liver. So those are just a few ways that while alcohol does produce a lot of energy for your body, overall it's still not good for you based on all the side effects it can produce. So now let's talk about what happens when glucose is low in the body. Essentially what will happen first is, since your brain always needs to be running, no matter what, is we will utilize, instead of glucose, something called ketone bodies, which we discussed a little bit before in beta oxidation. And essentially what these will do is these can be used in the brain to essentially be sort of energy substrates in the body to generate NADH, which can lead into the TCA cycle. The way this happens isn't exactly important. I would just really remember that ketone bodies are used in the brain. This is also kind of how a keto diet works, in the sense that your body isn't going to degrade your muscles right away. We're going to essentially turn acetyl-CoA into ketone bodies. And from there, instead of eating ourselves, essentially, we will use some of these energy stores from fatty acids before we degrade things like our muscles and just our other vital organs. Kind of common sense, but just a good thing to know. But since our body will always need glucose in some capacity, even when our energy is low, we can do something called gluconeogenesis. And essentially what this is this is creating glucose from non-glucose sources. And we have three things we like to do that with, essentially. You can do it with some more, but the biggest three you'll see are amino acids, specifically alanine, in the sense that it is very similar to pyruvate in like structure. We have lactate. We've already seen that a little bit, how lactate and pyruvate can be interconverted. And then we also have glycerol, which is a component of fatty acids. So essentially what we can do is, in a similar way that we had glycolysis, we can actually reverse the process because a lot of those enzymes are reversible. Now this will take a little bit more energy at the beginning steps. It essentially flips the ATP generating phase and the preparative phase, in the sense that we have the same enzymes up until around fructose 1,6-bisphosphate. So if you have your glycolysis pathway memorized, all these enzymes are the same, as well as the products. It's just they'll go in and out. The big thing is these two new ones. We have fructose 1,6-bisphosphatase at this step, and we have glucose 6-phosphatase to be converted into glucose. And you will need to remember these two. So I would definitely star that. I can almost guarantee you'll get a question on those somewhere. But essentially by turning the lactate and alanine into pyruvate, we can essentially reverse its role in the body, instead of going from something that generates energy to something that produces glucose. And this does have some regulation. One of the big things is the diagram before, in some of the earlier lectures. You may remember the oxaloacetate aspartate shuttle, which allows pyruvate or oxaloacetate to cross the membrane. Pyruvate itself can't cross the mitochondrial membrane, but through the structure, by actually going into the TCA cycle temporarily and then coming out, we can get it back into phosphoenol pyruvate. And that's sort of how we can get it through the cell or through the mitochondria in order to go back in the cytoplasm, where glycolysis and gluconeogenesis will occur. Okay, so to finish off this video, we're going to talk about kind of the regulation of gluconeogenesis. So as I mentioned before in the previous slides, you can see this process actually produces ATP when it goes back this way, through creating phosphates. And something else interesting that is good to note is when ATP has high, gluconeogenesis isn't going to occur as much, which makes sense when you think about it and kind of break it down. If energy is high, why do we need to be moving glucose to other cells, other parts of the body? So it's pretty self-explanatory in that way. There's also some other cellular processes that regulate kind of this whole process besides just gluconeogenesis. A big one is acetyl-CoA. As you can see down here, acetyl-CoA, obviously substrate in the TCA cycle made from pyruvate. When acetyl-CoA is present, it will actually upregulate pyruvate carboxylase, which will cause more pyruvate to eventually go through this process, leave as malate out of the cell. So this is like negative feedback, right? So even though acetyl-CoA will upregulate this, eventually pyruvate levels will decrease, and it'll kind of go back to a stable level. So that's one regulator. Another thing is pyruvate carboxykinase, which is PepsiK. That is actually inducible by glucagon. So you can see that glucagon increases adipose and other tissues to break down cells, or not cells, sorry, things like fatty acids into acetyl-CoA, which upregulates intracellular CAMP. You may remember that also from earlier glucagon signaling, which can essentially occur more regulation through that pyruvate carboxykinase that we mentioned before. So that's an upregulator there. And so basically, long story short, is glucagon will also move more pyruvate out of the mitochondria, which can regulate itself. And this is important because obviously if more pyruvate's out of the cell, more of it can undergo gluconeogenesis and get sugar out of the cell and get it where it needs to be. So with that, good luck on your quiz or exam, and I'll see you in the next one.